Two statement question. Statement 1 says antibiotics are chemicals, correct, produced by microbes, yes, that kill other microbes, correct. So penicillin is produced by penicillium, it inhibits staphylococci and it is called antibiotic. So statement 1 is correct. Statement 2 says antibodies are chemicals. They are proteins, but proteins ultimately are chemicals. So this is correct. Formed in body, yes, B lymphocytes produce it and they eliminate other microbes. Correct. So both statements are correct. So we will choose option number 3. Both statements are true. And this is the confirmed answer. So mark it on the OMR sheet 171.3. So this is the time taken to attempt this question. So see, statement questions are intricate questions. So you have to read it and analyze it thoroughly, every part of it, and then only mark your answer on the OMR sheet. So let us understand what antibiotics and antibodies are. So antibiotics is discussed in the chapter of class 12, Microbes in Human Welfare. And this is from a different chapter, which is Human Health and Diseases. So let's understand one by one. Antibiotics are chemical substances which are produced by some microbes and kill or retard growth of other microbes. So this point is very important, the other microbes. Now see, microorganisms include bacteria, it also includes fungi. Okay, so the initial discovery was, it was a chance discovery, serendipity. So Alexander Fleming, he observed that staphylococcus were inhibited by a mold. So later the chemical produced by the mold was named penicillin because the name of the mold is Penicillium notatum. The full potential was established by Chain and Florey. Very important. Once this was established, it was mass produced, commercially produced and used to treat infection, especially during war. And this is the reason why this discovery is so important, and all three were awarded Nobel Prize in 1945. So these are the details given in the NCERT textbook which you have to remember. So see, be careful about reading every statement of NCERT because this is how the questions are designed. The NEET paper is actually a test of how well you know your book. It is not the test of biology in general. It is about the content which is given in NCERT textbook. So that's why I have picked up all the important lines and compiled it here in this slide. Going on to the next topic, antibodies. So antibodies are produced by B lymphocytes and they produce an army of protein. That means a single B lymphocyte will produce lot of antibodies, thousands of molecules of antibodies in response to pathogen into our blood. This is very important. It is secreted. So antibodies are secreted in the blood. Therefore, you can do antigen antibody interactions using serum from the patient or any individual. Each antibody molecule has 4 peptide chains. Very important. Whenever you have association of polypeptides, it will be quaternary structure. So antibody is an example of quaternary structure. Okay, so you have to also keep in mind the ramifications of the content which is given to you. What all questions can be designed from the statements. So there are 2 light chains and therefore it is L2, and there are 2 heavy chains and it is represented as H2L2. So it will look something like this. This one is the heavy chain, 2 heavy chains and 2 light chains. And therefore this is also called as Y-shaped molecule. It looks like a Y. and represented as H2L2. Different types of antibodies are produced in our body, and these are the different classes: IgA, IgM, IgE, IgG. So the first statement says it is secreted in the blood, okay, and the response is called humoral. So this is in contrast to the response seen in by T cells, which is called cell mediated response or cellular immune response, and both come under adaptive immune response. So see, this is the image given in NCERT textbook, and it resembles Y. But you have to remember one thing, this is just the representation. The molecule actually looks something like this. So again, remember one thing, these structures, they will form a three-dimensional conformation, and that will fit in the structure of the antigen. Okay, so H2 and L2. So the heavy chain and light chain together will form the antigen binding site. So this is called the fragment antigen binding site. This region is called antigen binding. And this region is called the fragment which is constant. So the question which they can frame from this diagram is, so which chains are involved in forming the antigen binding site? So you will have to say both light chain and heavy chain, they together associate and they form a three-dimensional conformation. So this is how the conformation will look like. Okay, they will associate and form a 3D structure that fits in very well with the antigen. So antibodies are highly specific. They can identify small differences. For example, the AB blood group. So there is one single sugar difference between A and B, and that is also identified by antibodies. So this property of antibodies to identify small differences is called specificity, and the specificity is because of the antigen binding site. So this region is also the N terminal. Okay, remember one thing. See, they have written N over here and C over here. So N indicates N terminal. So N terminal is the first amino acid of the polypeptide. Polypeptide is a polymer. So the first amino acid will be called to be present in the N terminal region, and the last amino acid is at the C terminal region. So the question which they can frame is, antigen binding site is at the N terminal or C terminal. So you should be able to say the answer. Then what else? You can notice there are SS bonds. So SS bonds are called disulfide bond. So this helps in stabilizing the structure of the molecule. And what else? The types. So see, the constant region, this orange region and the red region, they are specific in the sequences, and they have a constant sequence. So light chain will be kappa or lambda. These two different sequences will be seen in the red region, and in the orange region you will see alpha or mu, which will give rise to IgM, then gamma will give rise to IgG, and epsilon will give rise to IgE. So I will not go into the details of this, it is not in your syllabus, but remember one thing, antibodies are like tags, let me just clear this. So you will have, if this is the pathogen, so the antibodies will bind to it. Okay, so this bound antibody will be a signal to other components of immune system like the complement system or the NK cells, okay, or the macrophages. They will get a hint that something which is unwanted is circulating in the body, in the blood, and it has to be eliminated or destroyed. So these other components will be triggered once the antibody binds to the pathogen. So this is how antibody works. And therefore, to communicate with these components of immune system, the region, FC region, has to be constant. Okay. So this will show a high degree of variety because it binds to different types of pathogens. See, you have whole range of bacteria. You have gram negative, gram positive, staphylococci, streptococci, and then there are E. coli, salmonella, shigella. So lot of variety of bacteria are there, but the mechanism of killing will be via these cells. So one end has to engage in variety of microorganisms and the other end has to maintain the constancy in the immune response. So this is an excellent example of high degree of variety and high degree of constancy in the same molecule. And this is the reason it took a lot of time for scientists to decipher and come down to this structure and finalize the structure of antibodies. So I will not go into the details of it. So what you need to remember is all these statements, and any type of question can be framed on it. So that's all about this question. Thank you so much for watching my video.